Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this video, I will be going over all the reactions of urea cycle and also regulation of urea cycle. Now let's get into details. Liver is the only organ where urea synthesis occurs, and urea is the major nitrogenous excretory product in humans. And urea, it accounts for 86% of nitrogen that is excreted from our body, and the rest of the nitrogen is eliminated as a creatinine, which accounts for 4.5% of the total nitrogen that is excreted. And it is excreted as ammonium ion in the urine, that is 2.8%. It is excreted as uric acid, that contributes to 1.7%, and rest 5% in the other compounds which are containing nitrogen. Like this, the major nitrogen containing product which is excreted out of our body is the urea, which accounts for 86% of nitrogen being eliminated out of our body. Approximately 30 gram of urea is excreted per day, and the amount of urea that is excreted, it depends on the protein intake. Higher the protein intake, more the urea synthesis and excretion. The reason for that is, whenever a more protein is consumed beyond the needs of our body, amino acids will undergo degradation, where the carbon skeleton will go into gluconeogenesis process, where it will make intermediates of glycolysis or TCA cycle, and they can be used for energy purpose or can go into fatty acid synthesis and can form glucose also. Whereas the nitrogen that is coming from that excess amino acids that a person consumes will go into urea cycle by transamination reaction. Ultimately, the amino groups will be given to glutamate. Glutamate undergoes glutamate dehydrogenase reaction to give a ammonium ion for urea cycle. And urea synthesized in the liver is transported in the blood and is excreted by the kidney. Now let's move on to see reactions of urea cycle. As you see here in the figure, urea cycle has 5 reactions. In that 5 reactions, 2 reactions are present in the matrix of mitochondria, and that is number 1 and number 2 reaction. That is number 3, number 4 and number 5 reaction. As you note that number 1 reaction in the urea cycle, which is present in the mitochondrial matrix, is a condensation or the fixation of ammonium ion to bicarbonate to make carbamyl phosphate. So in this process, 2 ATPs are consumed, where 2 ADPs are released and 1 inorganic phosphate is released, because the other inorganic phosphate coming from a terminal phosphate of ATP will be incorporated in carbamyl phosphate, which is the product of carbamyl phosphate synthetase 1 enzyme. And also note that carbamyl phosphate synthetase 1 enzyme, it is the regulated enzyme in urea cycle, and the molecule N-acetylglutamate, it acts as a positive modulator of carbamyl phosphate synthetase 1. We are going to look into the details of regulation of urea cycle in subsequent slides. Now let's focus on what happened to carbamyl phosphate in the matrix of mitochondria. Now the carbamyl phosphate is simply condenses with ornithine by an enzyme ornithine transcarbamylase, and this ornithine transcarbamylase, it is one of the mitochondrial enzyme participating in urea cycle, especially the second reaction. Not only that, the gene for ornithine transcarbamylase, it is located in the X chromosome. That's why if there is any deficiency in the ornithine transcarbamylase, it will give rise to an X-linked recessive condition, and that is called as ornithine transcarbamylase enzyme deficiency or OTC deficiency, ornithine transcarbamylase deficiency. So what is the product of this reaction? Carbamyl phosphate condensing with ornithine, it will make a citrulline, and this citrulline, it will move out of mitochondria into the cytoplasm while ornithine is entering in via an anti-porter system located in the inner mitochondrial membrane. Now once the citrulline is out in the cytoplasm, the citrulline is going to condense with aspartate to make argininosuccinate, and the reaction is catalyzed by argininosuccinate synthetase enzyme, and need to remember here ATP breaks into AMP plus PPR. That means equivalent of 2 ATPs are consumed in the reaction. So far, up to formation of argininosuccinate, we have seen 4 ATPs are consumed, 2 in the first reaction and 2 in the third reaction. Now the argininosuccinate is a complex molecule there. It is going to break into fumarate and arginine, done by argininosuccinate lyase enzyme. So we got arginine there, which is an amino acid itself. And that arginine, depending on the body conditions, especially in the liver, if the liver needs arginine for a new protein synthesis, arginine can be simply diverted into protein synthesis. At that time, there is no urea formation in the fifth reaction. And that happens in positive nitrogen balance condition where more and more proteins or new proteins are synthesized. So that is why urea levels in the urine will be down or decreased in positive nitrogen balance condition, because arginine is diverted into translation process. Otherwise, arginine breaks into urea and ornithine, and that is done by arginase enzyme, which adds water and break the molecule, which belongs to hydrolase class of enzyme. Now, one of the product of argininosuccinate lyase, which will break argininosuccinate into arginine, is a fumarate. This fumarate, it can go into TCA cycle, and in the TCA cycle, fumarate undergoes a TCA cycle reaction. Fumarate is converted to malate, malate to oxaloacetate, oxaloacetate. So like that, it will have a fate. We are going to see that later. Okay, so like this, number 1 to number 5 reactions are urea cycle reactions, which will make sure the ammonium ion coming from glutamate dehydrogenase reaction and the aspartate coming from glutamate transamination reaction done by aspartate transaminase, bringing the nitrogen. So they both are incorporated into urea formation. That's how urea is NH2CO NH2 there. Okay, in that NH2CO NH2, one of the nitrogen, so I am going to write that structure of urea here just above this, NH2CO NH2. So 2 nitrogens are there in the urea. One of the nitrogen is coming from ammonium ion, and the other nitrogen will come from aspartate. So that's how you can track 2 nitrogens that are present in urea. Now the source of carbon is a carbon dioxide or the bicarbonate. So I'm going to color that here. See, the bicarbonate carbon here is the one that you are going to find in urea molecule. So this is how we can track the atoms present in a urea molecule. So one of the nitrogen is coming from ammonium ion, second nitrogen is coming from aspartate, ultimately they both come from glutamate, you know that, and then the carbon will come from bicarbonate. So this is how a urea molecule goes. So in the urea cycle, these 5 reactions will make sure ammonium ion or the nitrogen, which is transported from all other amino acids from the peripheral tissues in the form of glutamate or glutamine, will become part of urea cycle and make urea. So that's how the urea cycle will help in neutralizing ammonium ion and excrete that nitrogen out of our body by the kidney into the urine. Now let's look at what is the fate of fumarate that has been released in the urea cycle. So the fumarate, it enters into the mitochondrial matrix and undergoes TCA cycle where fumarate converted to malate. Malate will move out of mitochondria to get into the cytoplasm, and in the cytoplasm malate is converted back into oxaloacetate, and oxaloacetate will form aspartate in transamination reaction, and that aspartate gets into urea cycle. So this type of link here between our TCA cycle and urea cycle is referred as Krebs bicycle. Now let's move on to see a regulation of urea cycle. So the urea cycle, it's a disposal pathway, meaning urea cycle is meant to excrete a nitrogen out of our body, especially the ammonium ion out of our body in the form of urea. So as like any other disposal pathways, so the availability of substrate is the one that will induce a urea cycle enzymes. So the substrate availability increases the activity of urea cycle enzyme, and also urea cycle enzymes can be regulated by at the gene level and also regulated at the enzyme level. So the gene level regulation, we can refer it as a course regulation where induction and repression going on, whereas the enzyme level regulation usually by allosteric modulation is a fine control. So whenever substrate availability is high, like especially if a person is in catabolic condition for a long period of time, like chronic disorders, so there will be 10 to 20 fold increase in urea cycle enzymes because protein catabolism is going on and urea formation has to be increased to handle that ammonium ion coming into the liver in the form of glutamate, because there will be higher glutamine levels in the blood because transamination reactions are going on at a higher rate. Fine control can be a regulation by the substrate availability, which is a feed forward regulation, and the other is regulation at the carbamoyl phosphate synthetase 1 enzyme done by N-acetylglutamate, which is an obligate activator of carbamoyl phosphate synthetase 1 enzyme. So what is the meaning of obligate activator? Obligate activator is the one that is absolutely necessary for the activity of an enzyme. That means in the absence of this obligate activator, enzyme generally doesn't function. So there are only two examples of obligate activators in our body. One is CPS-I enzyme that we are seeing now. The obligate activator for CPS-I is NAG, N-acetylglutamate. So the other is pyruvate carboxylase enzyme. And the obligate activator for pyruvate carboxylase is acetyl-CoA. Make sure you remember this. So these are the two enzymes that needs obligate activators in our body. CPS-I involved in urea cycle, very first reaction, which needs NAG as an obligate activator. Pyruvate carboxylase, which is there in gluconeogenesis process, conversion of pyruvate into oxaloacetate, that needs acetyl-CoA as an obligate activator. Now let's look at the role of N-acetylglutamate as an obligate activator on carbamoyl phosphate synthetase 1 enzyme. So this N-acetylglutamate, it is itself is synthesized by enzyme N-acetylglutamate synthase. And this N-acetylglutamate synthase itself is activated by arginine. And also note that N-acetylglutamate synthase, it needs glutamate and acetyl-CoA as the substrates. And if you think that when the conditions of increased availability of glutamate and acetyl-CoA in the cell is always a catabolic condition together. If there is high levels of glutamate, high levels of acetyl-CoA and high levels of arginine, usually it's a negative nitrogen balance condition and a catabolic condition. Because in negative nitrogen balance condition, there is increase in arginine, because arginine is not used for protein synthesis. More and more protein catabolism is going on in negative nitrogen balance condition, giving more arginine. And also when more and more proteins are undergoing degradation in the peripheral tissues, amino acids are undergoing transamination, making more glutamate, because product of transamination is glutamate, and glutamate level rises in the liver. So increase in arginine, increase in glutamate. And then the catabolic condition, when glucagon is more, adipose tissue lipolysis is going on, fatty acids are undergoing beta oxidation, and there will be more acetyl-CoA. So acetyl-CoA levels are more. So under catabolic condition, there is more arginine, more glutamate, more acetyl-CoA. All this makes a conducive environment to make N-acetylglutamate, done by N-acetylglutamate synthase enzyme. And N-acetylglutamate, now it acts as a positive modulator on carbamyl phosphate synthetase 1 enzyme. Thereby, carbamyl phosphate synthetase 1 is going to condense carbon dioxide with ammonium ion by consuming 2 ATPs to make carbamyl phosphate. And that's how urea cycle reaction is regulated in response to catabolic condition, because there is rise in glutamate, acetyl-CoA and arginine, making it favorable for synthesis of N-acetylglutamate, which is an obligate activator of carbamyl phosphate synthetase 1 enzyme. Now let's move on to see what is the relationship between gluconeogenesis and urea cycle. Note that gluconeogenesis goes hand in hand with urea cycle. Why? Because whenever skeletal muscles undergoing proteolysis, one of the important amino acid that comes out of the skeletal muscle is alanine along with the glutamine. In fact, compared to glutamine, alanine is the most predominant amino acid that comes out of skeletal muscle upon proteolysis. And this alanine, which is taken into the liver, will undergo a reversible transamination reaction to give its nitrogen to glutamate, while its carbons are there in the form of pyruvate. So I am going to color those carbons present in the pyruvate differently, and the nitrogen coming out of alanine in a yellow highlight that is present in glutamate there. So we are basically looking at the carbons of alanine and also nitrogen, amino group nitrogen present in the alanine. Now the amino group nitrogen present in the alanine will be present in glutamate after transamination reaction, whereas the carbons of alanine present in pyruvate. Now the pyruvate can undergo gluconeogenesis process in the liver. So glucose will be formed. So that means the carbons present in glucose are actually came from pyruvate, which in turn came from alanine. Since glucose has 6 carbons, alanine has only contribute 3 carbon making pyruvate. That means we need 2 pyruvates to make 1 glucose. That means we need 2 alanine to make 1 glucose. So I am going to add number 2 here. So 2 alanine making 2 pyruvate and 2 pyruvate going into 1 glucose, and 2 alanine making 2 glutamate, and in that one of the glutamate will be going into ammonium ion formation done by glutamate dehydrogenase reaction. So we have seen that already, glutamate dehydrogenase reaction removing amino group nitrogen as ammonium ion, and that ammonium ion go into urea cycle, contributing to one of the nitrogen in the urea. Now the second nitrogen present in second glutamate, which is undergoing transamination reaction with oxaloacetate to make aspartate. So the second nitrogen is there in the aspartate, and that will go into urea cycle and contributing to the second nitrogen of urea. So that means 2 alanines, when they undergo gluconeogenesis process in the liver, we are going to get 1 glucose, and for every 2 alanine giving 1 glucose, there will be 1 urea formation. That means for every glucose that is made in the liver, there will be formation of 1 urea molecules. For glucose to urea, 1 glucose to urea is 1:1 ratio during gluconeogenesis. So gluconeogenesis goes hand in hand with urea cycle. Okay, you got to remember that. That's the relationship between gluconeogenesis and urea cycle. So this is what is all about different reactions in urea cycle, regulation of urea cycle, and also relationship between urea cycle and gluconeogenesis. That's about it. I hope this video has helped you in understanding urea cycle and the details of regulation of urea cycle. Thank you.